Welcome to my Genetics 311 mini video. Today I'll be following the 4.1d prompt explaining the negative inducible control of the Lac Operon. The Lac Operon is a bacterial operon that encodes proteins that allow the bacteria to use lactose as an energy source. Here's an at-home model of the Lac Operon with a wild genotype. First we see the regulator gene LAC-I, which is transcribed and translated to produce regulator proteins. Further down we see the promoter region LAC-P, which binds RNA polymerase for transcription. The end of the promoter region and the start of the LAC-Z gene make up the operator region, which is responsible for binding any regulator proteins. The following three genes, LAC-Z, LAC-Y, and LAC-A, are structural genes, which are transcribed and translated to produce enzymes responsible for breaking down lactose. Since some bacteria metabolize multiple types of sugars, the Lac Operon is under negative inducible control, meaning it produces its own repressor protein to prevent the production of lactose metabolizing enzymes. The LAC-I gene codes for this repressor protein, which binds to the LAC-O site, blocking RNA polymerase from transcribing the structural genes, as seen here. When this repressor protein binds, RNA polymerase can't go through and transcribe the structural genes. So what happens when there's lactose to be metabolized? Allolactose is formed by the enzymatic conversion of lactose by beta-galactosidase. It binds to the regulator protein, causing an allosteric change and not allowing it to bind to the operator. With no repressor bound to the operator, transcription and translation can occur freely, producing enzymes beta-galactosidase, permease, and transacetylase. Mutations can also occur at the Lac Operon that disrupts this process. An I- mutation causes the repressor protein to be unable to bind to the operator, allowing the operon to be always on producing enzymes. An IS mutation does the opposite, keeping the operon off at all times since it cannot bind to the allolactase, and so it is always bound to the operator instead. A constitutive mutation in the operator prevents the repressor from binding, keeping the operon on. A mutation in the promoter keeps RNA polymerase from binding and thus prevents the operon from functioning. And lastly, if there are any mutations to the structural genes, then transcription and translation results in defective enzymes. All of these mutations can be hard to keep up with and contradict one another, so I've made a flowchart to determine if lactose metabolizing enzymes will be produced given a mutant Lac Operon. Feel free to pause the video and take a picture for future problems. So we'll look at this random diploid mutant Lac Operon. First we ask, what's a regulatory gene? Here we have IS and I-. IS is dominant to I-, so then we ask, what's our operator? We have a constitutive mutant for our O, OC. So then we go and we ask, what's our promoter? We have P+ and P+, so no mutations. We go right into asking, what are our structural genes? Our Z gene has one mutant and one normal, so we will make beta-galactosidase, but our Y gene are both mutant, so defective permease will be made.